DFG Science TV. Duck stop Bodensee. 260,000 waterbirds in search of food.
In recent weeks, Anja has captured birds for the isotope analysis. She would like to learn what the birds have eaten. To do this, she also needs samples of the food, of plants and mussels. It's windy today, perfect weather for sailing, but how will it be for collecting samples? Anja will look for the food at several locations around Lake Constance. Plants are certainly easier to capture than birds, but Anja needs a certain selection of these as well. The doctoral researcher and her assistant Gabi drop anchor. They scoop up plants out of the water and the mussels that have accumulated between them.
That's enough samples of the stoneworts. We'll put them into the cooler now.
On to the next sample location near Meersburg. The wind is picking up, the boat is rocking. The waves make the work more difficult. Anja is highly concentrated. Unfortunately, that doesn't help.
Still no mussels. We are going to have to go snorkeling.
In the lake, the zebra mussels, which are eaten primarily by the birds, cling to rocks. Unfortunately, these aren't caught by the anchor.
Yes, that should be enough in any case. We'll bag them now and then pack them in the cooler.
Particularly in sandy areas, it's best to search the few stones there are by hand. The life of a researcher can also have a more pleasant side. After completing their work, Anja and Gabi take a break. But there's still much to do. The samples need to be prepared for the analysis. To do this, Anja sorts them and determines their species. At 60 degrees Celsius, the plants, mussels and blood samples are dried out over the course of several days. For exact isotope analysis, the water needs to be completely evaporated from the samples. Less than 1/1000th of a gram of each sample needs to be weighed in tin capsules. A delicate touch is needed here. Not enough of the sample and the analysis is inexact. Too much and the analysis machine is overloaded. The feathers will be able to reveal precisely what the birds ate while the feathers were forming. The blood shows traces of the food from the last few weeks. The workflow. First, the capsule is weighed. Then a tiny piece of feather is cut off and inserted into the capsule. Everything is re-weighed and then carefully sealed. The sample is now ready for analysis. All that's missing now are just the last samples that need to be collected from the cages in the lake. Anja's last practical challenge before she evaluates her results.
Visit DFG Science TV for more information. Awaken the researcher within you.